Now let's take a look at the group B strep, the pneumococcus, and the oral streptococci. Group B strep are an interesting bunch. On paper, they look like they should be quite virulent. They should be a pretty serious threat. Remember, these are species, or pardon me, these are strains within the species Streptococcus agalactiae. They often carry hemolysins. Beta hemolytic is common, though it can be alpha hemolytic as well. What's scary is that they carry proteases and high hyaluronidase frequently. Proteases will chew up proteins, including things like collagen and connective tissue. And high hyaluronidases, we know what those do. Those chew through the extracellular matrix. And so group B strep, at least at first glance, looks like it ought to be a pretty invasive organism that would cause have a pretty high illness burden. They have a capsule as well, and the capsule is actually key to the reason why we don't get sick more often. It's not a hyaluronic acid capsule like we saw with group A strep, but it is a carbohydrate capsule, and our immune system in adults catches on quite quickly. So we readily detect it and we readily clear this pathogen. This is one that our immune system really has on the run. Now, by contrast, the infant immune system is not yet well developed, and infant immune systems don't do a great job at recognizing carbohydrate antigens in particular. That seems to take some time to develop. And so here you've got this group B strep coated in a carbohydrate capsule. And newborns in particular are highly susceptible to it. Now, when you add to that the fact that the only place we normally find group B strep in and on the human body is in the intestinal tract and in the female vaginal tract, you can see where we've potentially got a problem. The main disease that group B strep seems to cause is neonatal infections, neonatal bacteremia, meningitis, and even pneumonia. And it's because the baby passes through that birth canal and is exposed to the group B strep, which then its little infant immune system is not really well adapted to recognizing yet. And so these agalactiae bacteria have a great opportunity at spreading to some pretty scary locations like the bloodstream, the brain, and the lungs. Now, fortunately, in modern times, we have some pretty rapid tests, and so when a woman goes in for prenatal checkups, routinely we screen for group B strep, and if she's a group B strep positive carrier, perfectly healthy, then a note is made in her charts, and when she goes into labor, she's put on a suppressive antibiotic. And then when the baby is delivered, the baby is cleaned, and antibiotic eye drops, nose drops, mouth swabs, etc., are given to make absolutely sure that these group B strep bacteria don't transfer from the mother's birth canal to these sensitive locations in the infant. So since we've been able to detect group B strep over the last several decades, the incidence of these neonatal infections from group B strep has dropped dramatically, at least in developed countries like the US. It is still quite problematic in poorer parts of the world where the group B strep testing is not nearly as readily available. So then there's the pneumococcus. This little bugger causes a lot of problems. This is the most common bacterial cause of pneumonia, and that's where it gets its species name from. It's also the most common bacterial cause of ear infections, otitis media, in children, and it is a common cause of sinus infections as well. We'll talk about those infections in a minute. But the pneumococcus has four really important strategies, if you will, virulence factors that make it such a tough little bug to fight. So it's got a capsule, but from one strain of pneumococcus to the next, the capsule varies. There are over 90 unique polysaccharides now known, which is why we can't use the Lancefield system, because it's not consistent from one strain of pneumococcus to the next. And like any capsule, it's going to interfere with opsonization, interfere with phagocytosis in the same way that other capsules do. It makes it very slippery. It masks surface antigens and masks the ability of complement and antibodies to attach to the surface. Now, it also has, Strep pneumoniae also has a molecule on the surface called phosphorylcholine. And phosphorylcholine is a ligand, meaning a binding molecule. And it kind of acts like a long finger, and there's a doorbell at the other end. Our respiratory cells have choline receptors. That's the doorbell. The phosphorylcholine is the finger. And literally, pneumococcus attaches itself to the surface of our respiratory cells and rings the doorbell. Basically says, hey, pizza delivery, let me in, and it stimulates endocytosis. So they can actually be taken into your respiratory epithelial cells directly by having this phosphorylcholine on the surface. There are pili that are used for attachment, specifically to lung epithelial cells. And then it's got a hemolysin that we call pneumolysin, which is a very strong cytolytic exotoxin, and it punches holes in any of our cells. But in particular, because the pili and the phosphorylcholine are drawing these cells to our respiratory tract, or drawing these bacteria to our respiratory tract cells, the pneumolysin is really effective at punching holes in those respiratory tract epithelial cells. And when we do that, we lose the ciliary ladder. So think back from A&P, what the purpose of the ciliary ladder was. It's to move up and constantly move mucus and anything that we've inhaled, particulates, up and out. We clear our throat, and then we swallow it, and it goes down the digestive tract, and we destroy it. Without that ciliary ladder, we get a big buildup of mucus, and we become much more susceptible to pneumonia and secondary infections on top of the pneumococcal infection. So the diseases that the pneumococci can cause, sinus infection, middle ear infection, most common cause of bacterial pneumonia. There are cases of meningitis where it gets in the brain, bacteremia in the bloodstream. If it does get into the bloodstream, it has the opportunity then to also form an endocarditis by attaching to heart tissue and colonizing that heart tissue. Then we've got the oral streptococci. I mentioned earlier that these are also sometimes called Viridans streptococci. Very, very abundant in our mouths. They can be alpha hemolytic, on occasion they're gamma or non-hemolytic as well. These are the primary causes of dental caries and other oral diseases, gum disease and so on. They form biofilms on our teeth, and as those biofilms calcify, it forms that hard material we call plaque. And so the idea with brushing is to physically remove those biofilms before they can calcify and get hard, because when they calcify and get hard, it's still a live living biofilm. And remember, the streptococci are obligate fermenters, meaning that they're in there fermenting everything you give them, and they ferment it into entirely lactic acid. So here they are pinned against the enamel of your teeth. They're fermenting whatever food they can get a hold of, and they're producing acidic conditions right against the surface of your teeth, and that's going to etch away at that enamel and ultimately cause cavities. One cool thing about this is studies have shown that they require sugars, literally sugars like sucrose, to make their capsule, which means a low sugar diet should inhibit their ability to make their capsules, attach to your teeth, and form plaque. So lots of good reasons to cut back on the sugar. Now, before we leave the pneumococci and the oral streptococci, I want you to stop and think for a minute. The strep pyogenes, group A strep, and the pneumococcus often looks real similar. And in many cases, their signs and symptoms, their presentation are real similar. So learning how we can distinguish them can be really useful. All right, so answer these two questions. Make sure you can come up with a good articulate answer for both of these questions before you move on to the next video. So both group A strep and the pneumococcus are difficult to phagocytose. What virulence determinants does each pathogen possess that make this or mediate this resistance, that make this possible? Then ask the question, okay, both strep pyogenes and strep pneumoniae are able to lyse red blood cells and other cell types. What virulence determinants does each pathogen possess that mediate this attack? So make sure that you've got those big ideas clear before you move on to the next video.